Hello, my name is Mu Qiao, and today I'm going to present my postdoc work in Master Lab at Caltech, Factorized Linear Discriminant Analysis for Phenotype-Guided Representation Learning of Gene Expression Data. The motivation of this work is that it is important in neurobiology to relate the expression of genes of neuronal types to their phenotypes. And recent progress in characterizing neuronal types and investigating their gene expressions, especially with the help of single-cell RNA-seq, provides such an opportunity. The main problem here is that suppose we are given gene expressions of single neurons which are classified into cell types. We know the phenotypic traits of each neuronal type. Therefore, each type can be jointly defined by the phenotypic features. We want to find interpretable low-dimensional embedding in which certain dimensions represent factors of phenotype features or their interactions. We also want to recover these cell types, meaning that in the low-dimensional embedding, the cells of the same type are close to each other, while the cells of different types are far away. As a start, let's consider only two phenotypic features, I and J, both of which can be described with discrete categories. Each cell type can be jointly represented by features I and J, and the number of cells within each cell type is Nij. This representation can be described using a contingency table. Note here that we allow this table to be partial. Given gene expressions of each cell type, how to find linear projections that align with features I and J respectively. We came up with this method, factorized linear discriminant analysis, because the objective function has a similar format of that of linear discriminant analysis. Note here, NA equals MA minus lambda 1 MB minus lambda 2 MAB for a complete table. MA, MB, and MAB are the covariance matrices explained by the feature I, the feature J, and the interaction of them. Lambda 1 and lambda 2 are hyperparameters controlling the relative weights of MB and MAB with respect to MA. MB is the residue covariance matrix, representing noise in gene expressions. For the domain-specific application in neurobiology, we are interested in finding a small group of genes that best determine one of the phenotypic features. To do that, we impose sparseness on the number of genes contributing to each axis. Briefly, we solve this following optimization problem. This is a sparse generalized eigenvalue problem. We used the recently developed two-step algorithm specifically for this problem called RIFLE to solve it. We first tested our method on synthesized gene expressions to quantitatively compare it with other state-of-art methods in linear dimensionality reduction, such as principal component analysis, canonical correlation analysis, and linear discriminant analysis. Gene expressions of 4 cell types were generated from a Cartesian product of 2 features, I and J, organized in a 2x2 contingency table. We included the signal-to-noise ratio to measure how well each discriminant axis separates cell types compared with noise estimated from the variance within cell type clusters, and the modularity score to measure whether each discriminant axis depends on at most one feature. As shown in the plots, for both the signal-to-noise ratio and the modularity score, FLDA has their values close to the optimal value 1, because it not only considers alignment of axes to different features, but also constrains the variance within each cell type. To illustrate FLDA in analyzing single-cell RNA-seq dataset for real biological problems, we chose this dataset of Drosophila T4, T5 neurons. T4 and T5 neurons are very similar in terms of their general morphology and physiology, but they are different in their dendritic and axonal locations. Therefore, these cell types can be described by these 2 features, organized in a 2x4 contingency table. FLDA show that the axis corresponding to the dendritic feature and the first 2 components corresponding to the axonal feature have much higher signal-to-noise ratio than the remaining. We therefore project the data into the 3-dimensional space made of these 3 axes. Indeed, the 8 cell types are well separated in this space. We further extracted genes important to the dendritic and axonal phenotypes. Consistent with the previous report, we found indicator genes for dendritic locations such as TFAP2, DPR2, and indicator genes for the exon node targets such as KLG-BI. We also found genes that were not reported previously, such as PHCL1 for dendritic phenotype and LAC for axonal phenotype. They are promising targets to test in a follow-up study. To conclude, we developed FLDA, a novel supervised linear dimensionality reduction method, for understanding the relationship between high-dimensional gene expression patterns and cellular phenotypes. The sparse algorithm allowed us to identify new genes for each of the phenotypic features that were not apparent under conventional methods. The approach is motivated by multi-way ANOVA, and thus it generalizes easily to more than 2 features. The linear nature of FLDA makes it extremely easy to interpret the low-dimensional representations, as the weight vector directly informs the relative importance of each gene. Thank you for your attention.